Hi everyone, welcome back to my channel. I'm so happy to see you all here. So in this video, we are going to talk about some of the downstream analysis that we can perform with certain types of high throughput data, and we will be talking about that in further detail. But the highlight of today's topic is going to be motif discovery and performing transcription factor binding site analysis, looking for transcription factor binding sites, looking for motifs. But before we go into that, we are going to talk about what are motifs, what are transcription factors, how do transcription factor regulate or control the expression of genes, how are these motifs represented. More importantly, what kind of questions do transcription factor binding site analysis help to answer? And lastly, I would also want to perform a demonstration using one of the tools that is freely available online. I would want to perform motif discovery and also answer some specific questions with regards to a case study that we will be looking at. So without further ado, let's get started. A huge shout out to GeneXplain for sponsoring this video. GeneXplain is one of the worldwide known providers of top bioinformatics, systems biology, and cheminformatics databases, software, and services. They provide an extensive set of tools and comprehensive workflows to analyze high throughput data with a point and click interface. I will be talking in a little more detail about motif discovery and transcription factor binding site and performing these analysis using their platform and demonstrating their workflows and their tools in the upcoming videos. So please stay tuned. So let us start by talking about what are motifs. So a motif is a short sequence of nucleotides or amino acids that is repeated multiple times in a DNA, RNA, or protein molecule. And motifs can be as short as 2 nucleotides or as long as several dozen nucleotides or amino acids. And they're often found in specific locations in DNA or RNA molecules. So to give you an example, a very commonly studied motif is a Tata box motif that is found in the promoter regions of many genes. And a protein called as TFBS binds to the Tata box motif and which is involved in the initiation of transcription. Here in the diagram, you can see that there are many proteins that come together and bind to specific regions on the DNA in order for transcription to start. The Tata box binding protein is a transcription factor that binds to the Tata box in the promoter region, and this helps to recruit the RNA polymerase to the promoter region and initiate the transcription. So essentially there are two components that are at play here. So basically we have these specific patterns or recurring patterns in DNA that are called motifs, and to these motifs there are specific proteins that are binding which are called as transcription factors. So transcription factors bind to these motifs and they regulate the expression of genes. So going by this idea, it can be helpful to recognize these recurring patterns of DNA, these recurring motifs in the DNA, which can help us give us more idea on what kind of proteins or transcription factor bind to them and what sort of effects in terms of regulation of the gene occurs, and that can help us give more information on regulation of the expression of genes in different conditions. So motifs can be identified using a variety of computational methods, and one common approach is to search for sequences that are similar to known motifs. Another approach is to use statistical methods to identify sequences that are more likely to be motifs than random sequences. Now, these motifs play a very important role in different biological processes. Some of the most important functions of these motifs include gene regulation, as I previously mentioned, as these motifs are involved in the regulation of gene expression, like the Tata box motif to which the TF2 protein binds, and this results into the recruitment of other proteins and causes the initiation of transcription. Motifs have also been found to be involved in protein folding. So for example, the helix turn helix motif is a motif that is found in many proteins that bind to the DNA. And lastly, motifs are also found to be involved in protein-protein interactions. There are specific motifs that are found in proteins that interact with other proteins, and basically these motifs in such proteins help to mediate the binding of these proteins. Now that we've spoken about motifs, let's talk about the other component, that is the transcription factors. So transcription factors are proteins that control the rate of transcription of genetic information from DNA to messenger RNA. They do this by binding to specific sequences of DNA, and the binding of transcription factors to these specific DNA sequences can either stimulate or repress transcription. Transcription factors are essential for regulating gene expression, which is the process by which genes are turned on or off. Gene expression is tightly controlled in cells, and transcription factors play a key role in this process. So they allow cells to respond to different signals and environmental conditions, and they also allow cells to specialize in different functions. Now, there are many different types of transcription factors, and they can be classified into different groups based on their structure or function. Some of the most common types of transcription factors are activators. These are the transcription factors that stimulate transcription by binding to the promoter sequences and recruiting RNA polymerase, which is the enzyme that transcribes the DNA into RNA. There are something called as repressors, which are the transcription factors that represses transcription by binding to the promoter sequences and preventing the RNA polymerase from binding. Transcription factors can also be co-activators that work with other activators to stimulate transcription, which are other proteins basically to stimulate transcription. And co-repressors are the transcription factors that work with other repressor proteins to repress the transcription. The role of activator and repressor are context dependent for transcription factors. There are many cases where the same transcription factors can either be an activator or repressor in different genes and in different conditions. So depending on the cofactors, the context dependent role of transcription factor can be analyzed with transcription factor binding site analysis and understanding the combinations of various other cofactors that play a role in a certain process, which will be something that I will be talking about in a little more detail in the upcoming videos, where I go into the little more detail about the transcription factor binding site analysis. So here I wanted to give you a little bit more of an intuition or a visual representation of how the structure of a gene is and how the regulatory regions are organized. So essentially, genes are segments of DNA that contains the instructions to make proteins, and they are made up of three types of sequences. So the first are the exons, which are essentially the protein coding sequences, and they are transcribed into RNA and ultimately translated into proteins. We have something called as introns, essentially these lines between the exons, and these are non-coding sequences that are spliced out of the RNA before it is translated. And third, we have promoter regions, which are the regions of DNA that control the transcription of genes. So regulatory regions are sequences of DNA that control the expression of genes, and they can be located both downstream as well as upstream, or they can be present within the gene itself. Cis-regulatory elements are essentially these regulatory regions that are located in the same DNA molecule as the gene they regulate. There are also something called as trans-regulatory elements, which are these regulatory sequences that are present on a different DNA molecule from the gene that they regulate. So talking about promoters, promoters are the most important regulatory region in a gene, and they are located upstream of the transcription start site, and they contain sequences that bind to transcription factors, which are proteins that regulate the transcription. Now there is something called as enhancers, which are these regulatory regions that are located anywhere in the genome, but they are most often found upstream of the genes. And enhancers can increase or decrease the transcription of a gene, and it depends on the transcription factors that are binding to these enhancers. Now there are also other types of regulatory regions that are also found, and something called as silencers are these regulatory regions that decreases the transcription of a gene. They are most often found upstream of the genes, but they also can be located within the genes. Insulators are these other regulatory regions that can prevent the enhancers from activating genes that are located far away, and they are also found between the genes. And in addition to that, they can also help to ensure that the genes are only expressed in correct cells. So the whole idea behind painting this picture is to give you an idea as to how different regulatory region has a specific role. There are different transcription factors and cofactors binding to each of these regulatory regions, and together they all come together, coordinate to control the expression of genes. This complex system of regulation allows cells to respond to different signals and to produce the protein they need for functioning and survival. So in order to address a very important question that why are genes regulated, what is the need to regulate gene expression, we must understand that all cells within an organism share the same genome, which means that they possess the identical set of genes arranged in the same order along their chromosomes. While there are some exceptions, but this is not usually the case with most cells. And since most cells carry the same DNA, the key factor distinguishing one cell type from the other lies not in the genes they possess, but in which of these genes they activate or repress. This process involves transcribing specific genes into RNA and translating the RNA into proteins. So for example, a muscle cell differs significantly from a neuron due to the diverse range of RNAs and protein it contains. So muscle cell transcribe genes related to specific for muscle function like muscle actin and myosin, while neglecting the gene that encodes for neurofilament, which is typical for neuronal cells. So the decision of whether a gene gets transcribed or not largely depends on the presence of certain proteins within the cell, and this is something that we previously spoke about, that is transcription factors. So these transcription factors play a crucial role in regulating gene expression. The main control of gene expression happens during the initiation of transcription. So this process is regulated by core promoter elements and distant regulatory elements called enhancers, which we looked at in the previous slide. So additionally, there are other types of modifications like histone modifications and DNA methylation also play a vital role in regulating transcription, but we are not going to go into the details of that. If certain regions are inaccessible for transcription due to compacted chromatin or DNA methylation, the transcription process may not occur. In addition to that, gene activity can further be controlled by transcription. So non-coding RNAs like microRNAs and cell signaling can also influence gene expression by modifying the proteins or altering their interactions with other protein. So there are various levels at which the expression of gene is controlled, but here we are just looking at the transcriptional level by the transcription factors. So these modifications that occur after transcription or that are not occurring at the stage of the transcription are called as post-transcriptional mechanisms, which add another layer of regulation of gene expression. So just reiterating a little bit of what we spoke about in the previous slide, the transcription factors are the proteins that have the ability to recognize specific DNA sequences, and these factors can bind to different regulatory regions where they work together. They collectively regulate the expression of the gene and their transcription rate. So apart from their binding specificity, the concentration of these factors and the presence of other cofactors or cooperating or competing transcription factors can also play a role in determining the transcription rate of the gene. So let us look at different ways to represent a motif or a transcription factor binding site. So the first way you can represent a transcription factor binding site is by creating a consensus sequence. So by comparing the vast collection of well-defined sequence motifs, one can conduct searches for sequences or patterns and regions that exhibit sequence conservation. And these observed conserved regions or patterns can then be represented as a consensus sequence. The consensus sequence is defined as the most frequently occurring residues. They can be either nucleotides or amino acids at each position in the multiple sequence alignment. You will also be able to notice that in this consensus sequence that is generated from this multiple sequence alignment, there are additional one-letter codes that are not the standard A, T, C, and G. So here the Y represents that this position can have either a base C or a T, and H here indicates that this position can have a base A, C, or T. One can also create a position frequency matrix, which essentially shows the frequency of each nucleotide occurring at each position in the transcription factor binding site. The position frequency matrix is typically represented as a table where each row corresponds to a base, that is A, C, G, and T, and the numbers here indicate the frequency of that base occurring at that position. So to give you an example, the nucleotide A occurs twice at the third position, and it occurs 7 times at the fourth position. Now, using the position frequency matrix, one can create a position weight matrix, which is also called as position specific weight matrix, or also called as position specific scoring matrix. So essentially, position weight matrix is a matrix that typically represents the probability of each nucleotide occurring at each position in a motif. The values in a position weight matrix are typically represented as log odd scores, which means that they represent the difference in log odds between the probability of nucleotide occurring at a given position in a motif and the probability of it occurring at that position in a random DNA sequence. So essentially, the nucleotides are represented by letters A, C, G, and T, and the log odds scores represents the numbers are represented as numbers in the matrix. So essentially, the higher the log odds score, the more likely the nucleotide is to occur at that position. And finally, the consensus sequence, position frequency matrix, and position weight matrix can be represented as sequence logos. And these would be the logos you might have often seen while reading up a resource on motif analysis, as motifs are often represented as sequence logos. So sequence logos are a graphical representation of sequence alignments, position frequency matrix, or position weight matrix. And each logo consists of a stack of symbols. One stack corresponds to one position in the sequence. The overall height of the stack is proportional to the information content at that position, while the height of the symbol within the stack indicates the relative frequency of the amino acid or nucleic acid at that position. So in general, a sequence logo provides a richer and more precise description of, for example, a binding site than would a consensus sequence. The information content in sequence logos is measured in bits, and in case of DNA, it ranges from 0 to 2 bits. So essentially, the idea here is that a position in the motif in which all the nucleotides occur with equal probability has information content of 0 bits or close to 0 bits, and while a position at which only a single nucleotide can occur has high information bit or near to 2 information bits. So basically, positions that are highly conserved will correspond to high information content, while positions that are not highly conserved will correspond to low information content. It is important to understand what kind of questions do transcription factor binding site analysis can help to answer, as you would only want to pick this analysis and do these analysis if you are asking some of the questions written here, or you're asking the questions on the lines of these questions I've provided here. So broadly, there are two types of analysis that can be performed. So the first is motif discovery, which is essentially the process of identifying short conserved sequences of nucleotide that are likely to be binding sites for transcription factors, and this is done by searching through a large data set of DNA sequences, or there are also motif databases that are available, and identifying the sequences that have a high degree of similarity to them. And this can be performed to find de novo motifs or motifs which have been previously identified and studied. You would essentially want to perform a motif discovery if you're asking questions like what are the binding preferences of a particular transcription factor, whether it's a known motif that you're trying to study. You could also be asking how do transcription factors regulate gene expression in a particular disease or a condition, and how does DNA sequence variation affect transcription factor binding. If you are asking any of these questions or questions on these lines, then you would want to perform a motif discovery. The other type of analysis is motif enrichment, which is essentially the process of comparing the distribution of particular motif in a set of target sequences to its distribution in a background set of sequences. So basically, you would essentially want to see whether there are certain motifs that are enriched in your regions of interest or in your targets, and this can be used to identify genes that are likely to be regulated by a particular transcription factor. So if you're asking questions like what genes are regulated by a particular transcription factor, how do motifs contribute to the regulation of gene expression in different cell types or under different conditions, or how do motifs contribute to development of diseases, or how can transcription factors be targeted for drug development, or any questions on such lines, then you can perform motif enrichment. This is to say that it is possible to perform both these analyses together. You can also perform motif discovery and find whether the motifs that you find are enriched in your regions of interest. It is possible. In addition to that, there are also several other analyses that can be performed. So this is just a very broad classification and a very simplified classification, but there are a lot more analysis and combinatorial approaches that can be taken. So here I am listing several tools and web platforms that are available to perform transcription factor binding site analysis, and these tools use various algorithms and techniques to analyze ChIP sequencing data, DNA sequencing, as well as other experimental data to identify transcription factor binding site and motifs, and also some related analysis. So here we have Homer, which is essentially a widely used motif discovery and transcription factor binding site analysis tool. It can identify enriched transcription factor binding site from DNA sequences and chip sequencing data. And today I will be using this tool to demonstrate some transcription factor binding site analysis. In addition to that, we have Meme Suite, which is essentially a collection of tools for motif discovery and transcription factor binding analysis. We also have Jasper. Jasper has both the database, the transcription factor database, as well as tools to perform motif analysis. There are also these two gigantic tools, which is GeneXplain's platform and Genome Enhancer by GeneXplain, which essentially provides end-to-end workflows and tools using point-and-click interface without the need of any programming to perform and analyze your data, as well as generate insights and perform motif analysis. Genome Enhancer particularly also allows us to identify master regulators, and I will be talking more about that in my upcoming video. So here I'm just listing out some of the very popular motif databases which could be a valuable resource for researchers studying gene regulation and transcription factor binding. These databases contain experimentally validated motifs as well as computationally predicted binding sites, facilitating the identification and analysis of regulatory elements in various species. So for today's demonstration, the goal of the analysis is going to be identifying DNA motifs, both de novo and canonical, that are enriched in genomic regions where RUNX1 binds. RUNX1 is essentially a transcription factor that plays a critical role in the development of breast cancer. And it has been reported that overexpression of RUNX1 can lead to activation of genes that promote cancer growth and survival. So for today's demonstration, the data that we are going to use is a ChIP-Seq dataset, and these there are essentially two samples that are generated from MCF10A cells. One is an input control, and one contains the region with RUNX1 where RUNX1 binds. MCF10A cells are essentially non-tumorigenic epithelial. These are the cells that are derived from the non-tumorigenic epithelial cells, and these cells are often used as a model to study the normal mammary gland development, as well as various aspects of breast cancer progression and cell biology. So for anyone who hasn't heard about or know much about chip sequencing or ChIP-Seq, how the data is represented in ChIP-Seq files, as today we are going to deal with a peak file. I have previously created two videos talking about, in one of the video I give an overview of different types of high throughput data, in which I broadly introduce chip sequencing, and in one of the recent videos I speak more about various file formats associated with chip sequencing. So I recommend you to watch those videos before you follow with me along in this demonstration, and I will be adding the link to both of those videos in the description section below. So let us first start by downloading the dataset, and I will go to NCBI GEO to download our dataset. So the accession ID for the dataset is GSE 129314. And this is a chip sequencing dataset, and as I said, the cells are MCF10A cells, and MCF10A cell line is essentially a non-tumorigenic cell line. So essentially this analysis is going to give us an overview of all the other proteins or transcription factors that bind to the regions where RUNX1 binds, and since these cells are from non-tumorigenic cell line, this would be a situation or like an overview landscape of proteins that are binding to these regions in a situation when cancer has not developed, since these cells are non-cancerous cells. In addition to that, I also want to make a point that we may find a lot of other interesting transcription factors binding to these regions, but binding of transcription factors may not always translate into regulation or interaction with RUNX1. So we may find a lot of other transcription factors, but regulation of a gene is context dependent, and there are a lot of other factors that play a role in regulation. So just something that we need to keep in mind when we are analyzing or looking at the results of motif analysis. So looking at the record here, as I said before, we have two samples. One is an input control and one contains the regions where RUNX1 binds. So if you click on either of the sample and if you scroll down, there will be a section called data processing. So essentially this section provides you with the details of how the data was processed. You'll get information on how base calling was performed, how reads were aligned, what genomic build was used, what tool was used for alignment. In addition to that, since this is a ChIP-Seq data, peaks were called using Max algorithm, and this was the command that was used to call the peaks. From the command, we can see the Q value cutoff set as 0.05. So all the resulting peaks would pass this cutoff value. And ultimately, the information that they provided is essentially the peak file. So basically, that has information on the peaks. So going back, we can download the peak file because we will be using this peak file. We'll be providing the information from this peak file to the Homer and call the motifs in the regions where RUNX1 binds. So you can download this peak file by clicking on the HTTP, and you can just download it onto your system. I have already downloaded this peak file, so I'm not going to do it again. So before I start with the demonstration, there are a few things I want to talk about Homer. So this is essentially the Homer website. Homer is a differential motif discovery algorithm, and it uses a set of background sequences as control to essentially find enriched motifs in your target regions or sequences. So Homer provides extensive documentation on downloading, installation, as well as running specific analysis. So before I jump into the analysis, I just want to make a note that a lot of you reach out to me asking me to create videos on installation and showing you how to install certain packages. The biggest challenge for me is to create a sort of a generalized video for installation, as everyone has a very different system setting, and it is nearly impossible for me to create or replicate these specific system configurations, as my system and the settings would be very different from any individual person's system or setting. So one way to deal with the package installation issues or conflicts or dependencies is to use environments. So I will definitely plan to create a video on Conda environments and R environments in the future, but in the meantime you can look that up, and if that is something that you would want to go ahead and venture out and try to install your packages and tools in environments, I think that is one of the very efficient way to go about this. So having spoken about environments, before we get onto that, here on the download page of Homer, you will get instructions on your system requirements, both hardware and software. You can download this file, and there are instructions on how to install Homer on your system. Once you have downloaded Homer, there are ways to install it. So there are installation instructions, and since we spoke about environments, there are Conda instructions to download some of the essential tools that are required, and you can create an environment and install Homer in your environment. So that will prevent package conflicts with already existing package with different versions on your system. There are also instructions on downloading and installing Homer in different systems. Their documentation is very comprehensive, so I don't think you should have a problem installing this onto your system. It was very straightforward for me to install Homer on my system. Once you've installed Homer, next you can go scroll down, and you can see there are a lot of tutorials that are available for various types of analysis. So I'm going to click on the Homer motif finding tutorial. So when I click on that, essentially it opens up a page which shows me a list of scripts or analysis that I can perform. So whether I have a list of genes or promoters, or whether I have genomic positions, or whether I have FASTQ files, there are different scripts that I can run and perform different analysis. And since the data that we are dealing with today is a ChIP sequencing dataset, which essentially provides us with regions where RUNX1 binds, and the goal of our analysis is to find the transcription factor and binding sites in those regions where RUNX1 binds, I'm going to click on the second one, analyzing genomic positions. So essentially this page provides me with a detailed and comprehensive description of how to use these scripts and what are the parameters that I need to provide. Additionally, they also provide information on the acceptable input files, providing me with what columns should hold what information. Scrolling further down, it also provides me a list of or a bullet points of all the steps this script performs to determine the de novo and the known motifs and the enrichment of these motifs in my regions of interest. And finally, they also provide a full description of all the output files that I will be getting and what information will be contained in each of those output files. And lastly, there are additional parameters and tips for motif finding, which can be looked at if there are any specific, if you want to tweak certain parameters or would want to add certain parameters, this would provide you with a detailed description of all those parameters. So in order to run this script, I would need a peak or a bed file. I would need to specify the genome. I would need to specify an output directory, and I will need to provide the size parameter. I'm just going to scroll down for the acceptable input files, and here either I can provide the peak file or I can provide the bed file, or Homer will accept a bed file with only 4 columns. So basically these 4 columns. They do not recommend running Homer with only the first 3 columns, that is the chromosome, the starting and the end position, because if you were to find which motifs occur in what peaks, you would not be able to do that if you do not provide unique peak IDs, because it cannot trace back that information. And all of this will make more sense once I demonstrate how to run this script and what information we get. So for the first thing that I'm going to do is that I'm going to explore my data and try to get my data into this acceptable file format. So I'm going to be providing a bed file with these 4 columns. I'm going to try to get my data in this format. So I'm going to keep this page open, and for now I'm going to minimize it and go to my terminal. And before I look at my data, I just want to quickly show you that I have 3 folders and my data is present in the data folder. So let's go to my data folder and let's take a look at this file. So essentially this is the peak file. There is some information provided here, all the arguments list and all the details about the file. And when I scroll down, I finally get to see the regions where Annex 1 binds. So chromosome start, end, the length of the region, and all the related information from the peak calling. So in order to provide this information to Homer, to that script, I need to get the chromosome start, end, and the 4th column should be the name, that is the peak IDs. So I need to manipulate this file a bit so that it's in a format that is acceptable by Homer and that can be run with Homer. So essentially there are 2 things I'm going to do here. I'm going to get rid of all these lines, and then I'm going to pick out the columns of interest. So the first thing I'm going to do is I'm going to find out the row number for this header line, and then I'm going to use a head or a tail command to get all the rows following this header line, and then I'm going to pick out the columns of interest. And let me show you how we do that using one line code in command line. So the first thing I'm going to do is I'm going to grab the row number for that header line. So the header line is at row number 30. So this is essentially the first row, that is the row 30th. So what I'm going to do is I'm going to use the tail command to get everything after the row 30th, including the row 30th. So as you can see that we've got rid of all the lines above. And once we do that, I'm going to use a cut command and choose the first 3 columns, and let me count the number of columns to get, so that I know which columns I need to pick. So this is column 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So I need columns 1 to 3 and column 10. So let's use the cut command and provide the columns that we need. And before we save this into another file, let's take a quick look. So this is exactly the format I need my columns to be in. So I need the first 3 columns to be the chromosome start and end positions, and the 4th column should be the unique peak IDs. And this should be the acceptable format that I can provide to Homer. So now let us save this into a new file called runx ChIP-Seq. peaks dot text, or let's put it peaks homer dot text. So now that you can see that we have manipulated and subsetted our data and saved the data into a new file, and this is the file that we will be providing to Homer. So now let us run the find motifs genome script from Homer. So I'm just going to open up the document again, and here I'm just going to copy the sample command, and I'm going to be replacing that with just doing an ls so I know what files are, and I'm just going to paste the command, and here I'm going to change the values. So we need to give it the peak file that we saved. The genome will be at G38. The output directory would be the results directory, which is one level up, and size would be, let me go back and refer to that. So I'm just going to go with the default size of 200. So basically the size here is the size of the region for motif finding, and the default is 200, and I'm going with the default. They did provide with some information about that, so selecting the size of the region for motif finding, and you can find the information here on the recommended sizes, but for this demonstration I'm just going to go with the default size of 200, and let us run this. Now that Homer has finished running, let us go back to the results folder and take a look at all the files that are generated. So here all the files that are starting with Homer motifs, and there is a number. These files essentially hold the results of the de novo motifs. So basically all the de novo motifs that has been found, and these represent separate runs of the algorithm, and these files are separated by the number of the k-mers or the motifs that were searched for. The Homer motifs.all.motifs file essentially contains all these motifs compiled in one file. Next we have Homer results directory. This directory holds motif files that were used to find specific instances of each motif. Homer results.html is the HTML report of the result of the canonical motif finding, and we will be shortly taking a look at this HTML file. Next we have known results directory. This directory holds the motif files that were used in finding specific instances of each motif, and that will make more sense once we take a look at these reports. Known results.html is another HTML report which holds the results of canonical motif finding. So basically all the files starting with Homer motifs or Homer results holds the information on de novo motif finding, and the known results indicate the canonical motif finding that is performed by Homer. Known results.txt holds the statistics associated with the canonical motifs that are found. Motif finding parameters, this text file holds the command that is used to execute this script. And this file essentially is, so basically there is some autonormalization step that is performed by Homer to account for the bias that is introduced by the shorter oligonucleotides. So basically this file contains the autonormalization statistics. So let us open these HTML files and take a look at these reports. So let us open these files in the browser. So first taking a look at Homer de novo motif results. So basically this gives, at the top of the report you will be able to see the total number of target sequences and the number of background sequences. So basically Homer tries to find the known motif with the best correlation between the known motif and the de novo motif, and it also provides the alignment of the de novo motif with the known motif. However, these results have to be taken with a grain of salt, as the top result or the top match may not always be the best match or may not always be annotated correctly. So these results have to be taken with a grain of salt and not to be taken literally. So here we are provided with a table of rank and the de novo motifs, the p-values, the log p-values, the percentage of the target sequences in which this motif has been found, the percentage of the background sequences where this motif has been found. This is essentially the distance or rather the average position of this motif in the target and in the background. And here is the best match details. As I said, the best match may not always be the best match, and you can look at the alignment and take a judgment call for you, and this may also require further investigation. And this is essentially, the last column provides a link to the Motif file, which is essentially the matrix. So let us take a look at the first one, which is the runx. And this is somewhat believable, as we are dealing with a chip sequencing data that captures the genomic locations where runx1 binds. So essentially the best match is a motif which is bound by runx. But let us click on more information link to get some more information on this motif. So here again you get a bunch of stats associated with your de novo. They also provide you with the sequence logo for forward and reverse opposite. And this is essentially a list of the matches to the known motifs, and this is the alignment that I was talking about. So this information can be obtained, and they also provide the match rank, scores, and the orientation for the matches. So basically this information can be used to determine how well a de novo motif matches with your known motif and whether any further investigation is required. And similarly, that information can be obtained for all the other de novo motifs that have been identified. There are also small red asterisks that could be present next to the ranks, which could indicate false positives, possible false positives. So you also need to be look out for that information if any of the motif that is a high scoring or has a extremely low p-value should not be a possible false positive detected by this algorithm. Now switching over to the known results. So basically known motif enrichment results provide us with the known motifs that are found to be enriched in my regions of interest, the genomic regions I provided. So here again at the top of the report we do get the total number of target sequences and the background sequences. Like the de novo results, we get the motifs by ranks. We have here the motif symbols, the name of these motifs, the p-values, the log p-values, the corrected p-values. We also get the number of target sequences where this motif was found. We also the percentage value for that in the target sequences calculated. Similarly, we get that information for the background sequences. We have the Motif file, just like what we saw with the de novo results. This is the matrix file for the motif, and we also get the SVG for this logo. So just as I said previously, I should expect to see a very strong enrichment of the DNA binding sites, or these motifs that are recognized by the transcription factor that is essentially being studied in my dataset. So basically, my dataset captures the runx1 binding sites. So I should expect, and this is what essentially I expect to see, that the motifs recognized by runx1 are amongst the top ranking motifs and the transcription factors. Had it been that I did not observe this, it would indicate that my ChIP-Seq experiment failed somehow, or my peaks are of low quality. So this should be a very good sanity check in terms of making sure your experiment ran successfully. If you do see a strong enrichment of the DNA motifs that is recognized by the transcription factor that is being immunoprecipitated by your ChIP sequencing experiment. So scrolling further down, I do see motifs that are enriched, which are bound by certain transcription factors, even the ETS family transcription factors, which have been previously reported to cooperate with runx1 for gene regulation. So dysregulation of these interactions could potentially contribute to disease progression in breast cancer. So essentially this provides us with the motifs and the transcription factor that binds to these motifs enriched in the regions where runx1 binds. And you can see that these motifs are highly significant motifs by looking at the p-values. So while scrolling further down, I am intrigued by seeing p53 here, which is ranked at number 41 here. So p53 is a transcription factor and it acts as a tumor suppressor protein, and it regulates cell division, so it prevents the cells from growing and proliferating uncontrollably. So essentially, I'm interested in finding which are the peaks where p53 bind to, essentially finding where this motif is enriched in the genomic regions I provided. So in order to do that, let us go back to the Homer documentation, and scrolling down, here they have provided ways to find instances of specific motifs. So basically there are two ways. We can use the current script and use this parameter and provide a motif file in order to find the motif instances, and the output file will contain these columns. And the second way is to annotate peaks and provide it with this parameter. So basically this would be the command to run annotate peaks and get the motif instances along with peak annotation. So I will be demonstrating both ways just to show you the output that is generated and how we could get motif instances and find out where this motif essentially is in what peak regions. So let us start with the first method. So let us use this example command and replace these values with our file names. So going back to the terminal. So let me paste the command and replace each of these values. So the peak file was present in data. We replace the genome with HG38. The output directory would be the current directory because we are in the results directory right now. And for the motif, so quickly going back to the result, so this is essentially motif number 41. So, and since this is the result from the known motifs, let's go into the known results folder, and known 41 would be the motif, and the motif file that we need to provide should end with .motif, and the output should be .text, and let's run this. So now this is finished running. Let us take a look at the results. So basically this is the file that was created, and just making this a little smaller so I could fit that in one window. So the first column is the position, the peak where the motif is found. The offset is essentially the distance from the center of the region. This is the sequence of the site. This is the motif name. So this was the motif that we were searching for. Next we have the strand information, on which strand did we find this motif, and the motif score. So basically these are the log scores. The higher the score, the better are the matches. So here essentially you will be able to see which peaks have your motif of interest, and you can also look at the offsite to get an idea on where this motif is located from the center of the peak. So now let us use the next method, the annotate peaks method, and let's go back to the Homer documentation and copy the example command, and we will be replacing this with our files. So basically here we provide the peak information, we change this to HG38, and here we provide the motif file. Again, this is going to be in known result, known 41.motif, and the output would be motif instances using annotate peak method .text. So now this is finished running. Let us just take a look at this file, and this seems like a very big file. So apparently there are a lot of columns which I would not be able to fit in this video. So let me open this file in Excel so perhaps we can look at the information better. So opening this information in Excel will allow us to view a lot of information in a more clear format. So here you will see there is a bunch of information associated with the regions in terms of the genomic location strand. This function basically provides the location to the peak in terms of the genomic features. So you get to know which peaks are in the intergenic regions, in the intron, or in the protein coding regions, or whether they are in the 3' or 5' UTRs. You'll also have detailed annotation. You have distance to transcription stat site and the nearest promoter, the IDs for that. You'll also find the gene names, the nearest ensemble IDs, and the gene names, their aliases, description, bunch of information. But I think our interest is in the last column, which basically provides where these enriched motifs are in which of these peaks. So essentially this column provides us with the information on the offset, the sequences, the strand, and the conservation score. So you can perform like a bunch of filtering on this data. You can also see in which regions do these enriched DNA motifs where p53 bind are observed, whether they are in the protein coding regions, whether they are in intronic regions or intergenic regions. So basically this information is more comprehensive in terms of filtering and looking for where your motifs are or where rather your transcription factors are binding. So that brings me to the end of this video. I hope you found this video helpful and informative. All the commands that I ran today, the homework commands, I will be putting it into a bash script, and I will be uploading the script to my GitHub repository, the link of which will be added in the description section below. I want to thank GeneXplain again for sponsoring this video, and I am looking forward to performing these analyses on their platform and using their workflows and tools in the upcoming videos. So please stay tuned. So if you found this video helpful and informative, please make sure you hit the subscribe button, like the video, share it, and leave your comments under the comment section. Until next time, see you.